Assalamualaikum. Now we are here with FSC Biology Chapter 4. This chapter is most important because almost 4 to 6 MCQs have already been seen in the past paper of lecturer and subject specialist. So, your patience to stay with me will inshallah help you to cover all aspects in these topics. Please watch online. In this chapter, we shall discuss first 10 topics of cell, while other remaining 10 will be discussed in the next video, inshallah. Links of previous 3 chapters which we have been discussed so far are given in the description. Cell theory. What is cell? Cell is structure and functional unit of life. Robert Hooke discovered cells in 1665 and published the work in Micrographia. Now these two keywords are important, Micrographia and 1665. He worked on section of cork, which are dead plant material. Robert Hooke only described cell as empty bones with thick walls because he observed plant cells. German scientist Lorenz Oken in 1805 used the term vesicles for cells. Lamarck had same idea and said no body can have life without cellular tissue. In 1831, Robert Brown discovered nucleus. Theodor Schwann, a German botanist and children zoologist, proposed cell theory. So, if we are asked who proposed cell theory, so these two names are kept in mind, Theodor Schwann and Sheldon. They recognize 3 parts of cell as nucleus, fluid and outer covering. Nucleus, fluid means cytoplasm and outer covering means plasma membrane or cell membrane. Rudolf Virchow in 1855 said omnius cellula is cellula in German. Means new cell originate from parent cell or already existing cells. Abiogenesis, which is a theory proposed that living things arise from non-living things, not the living things. So, the theory omnius cellula is cellula proposed a QoS in the minds of scientists. Rudolf Virchow and Louis Pasteur rejected this famous theory of abiogenesis. Louis Pasteur in 1862 experimentally proved this theory wrong, that the living things cannot arise from non-living things or non-living parts. In 1880, August Weismann said that all living things have common origin. This statement is very important to remember. Now, sclerenchymatous cell provides spore, collenchymatous cell carry out photosynthesis, parenchymatous cell store surplus food, meristematic cell result in growth and development, or the meristematic cells are responsible for growth and development in plants. Resolution of human eye is 1 mm. Resolution of compound microscope is 2 micrometer. Resolution of electron microscope is 2 to 4 angstrom. These are very important to remember. Now, how to calculate magnification power or magnifying power? Simply multiplying ocular lens and objective lens. Cells in general. Cell fractionation is used to study components of cells. Tissues are homogenized or mixed and mixture is centrifuged to separate components. High-speed centrifugation is called ultracentrifugation. Protoplasm is a complete cell except outer boundary. Cytoplasm is in between nuclear membrane and plasma membrane. Sizes of cells are measured in micrometers. 1 micrometer is equal to 0.000001 meter. Ostrich egg is the biggest cell. Now, plasma membrane. Now, your attention must be deep. In plasma membrane, 60 to 80% are protein, while 20 to 40% are lipids. According to unit membrane model, there are 2 models of plasma membrane: unit membrane model and fluid mosaic model. According to unit membrane model, membrane is lipid bilayer sandwiched between inner and outer protein layer. According to fluid mosaic model, proteins are embedded in lipid bilayer. In unit membrane model, lipid bilayer is sandwiched between inner and outer protein. But in fluid mosaic model, proteins are embedded like nails in the wood in lipid bilayer. Lipid soluble substance and neutral substance cross the membrane easily. Active transport through membrane requires energy, and that energy is provided by ATP. Taken in material in the form of vacuoles by unfolding is called endocytosis, and if engulfed material is liquid, it is pinocytosis. Remember that if engulfed material is liquid, it is pinocytosis, and if engulfed material is solid, it is phagocytosis. Now cell plasma membrane transmit impulses. Cell wall. Cell wall is secreted by protoplasm of plant cell. Cell wall has three main layers, which are primary, secondary, and middle lamella. Middle lamella is first to be formed between primary layers of neighbors. Primary wall is composed of cellulose, pectin, and hemicellulose in crisscross. Primary wall is true wall which develop in young growing cells, or you can also say that the first wall which develop in growing plant is the primary wall. Secondary wall is rigid and thick and is formed inner to primary wall. Chemically, secondary wall is inorganic salts, silica, waxes, cutin, and lignin. Prokaryotic cell wall lacks cellulose but peptidoglycan or murines. Cell wall of fungi contain chitin. Major functions of cell wall involve providing definite shape and rigidity. It does not act as barrier. Means it provides rigidity and shape to the plant cell, but it does not act as barrier like plasma membrane. Cytoplasm. Living content of cells are cytoplasm and nucleus. Soluble part of cytoplasm is called cytosol, which is a ground substance. It has 90% water. In cytosol, small molecule form true solution while larger one form colloidal solutions. If colloidal solution is non-viscous, it is called as sol, and if it is viscous, it is gel. Peripheral parts of plants are gel. Cytoplasm is storehouse and glycolysis occur in cytoplasm. Mitochondria are freely moving organisms in cell. Endoplasmic reticulum. Endoplasmic reticulum is the system of channels or tubes or tubule in cytoplasm throughout, and it may spread from plasma membrane to nuclear membrane. The spherical or tubular membranes of endoplasmic reticulum which separate material from cytoplasm are called cisternae. Rough endoplasmic reticulum has ribosomes, and thus they are site for protein synthesis, and they are site where protein are synthesized, and these protein are then pushed to Golgi apparatus. Smooth endoplasmic reticulum help in metabolism of lipids, detoxification of drugs, and transport. These are three main functions which smooth endoplasmic reticulum perform: metabolism of lipid, detoxification of drugs, and transport. In muscles and nerve cells, endoplasmic reticulum transmit impulses. Endoplasmic reticulum also provide mechanical support to maintain shape of cell. Very important point to note and remember. Please keep in mind. Ribosomes. Plaid was the first person to study ribosomes in 1955. Ribosomes are teeny granular structure found in cytoplasm and on rough endoplasmic reticulum. They are composed of equal amount of RNA and proteins. That's why they are called as ribonucleoproteins. They consist of two subunits. Larger one sediment as 60S Swedberg unit, while smaller at 40S. When attached together, they are ATS. Magnesium ions control attachment of larger subunit with the smaller one. Ribosomes attach with messenger RNA through small subunits. So, messenger RNA attach with ribosome with the help of small subunits. Group of ribosome attached to messenger RNA is called polysome. New ribosomes are assembled in nucleus, which is factory of ribosome. Factory for protein synthesis is also ribosomes. These are very important facts to remember. Please keep in mind and read in depth. Next is Golgi apparatus, which is discovered by Camillo Golgi in 1898 and found in all eukaryotic cell. Please remember that only eukaryotic cell, not prokaryotic. Stacks of flattened membrane-bound sacs are called cisternae. Cisternae attached with vesicles are called Golgi complex. These are very important definitions and have already been asked in PPSC exams. Cisternae stacks are formed by fusion of vesicles from smooth endoplasmic reticulum. Outer convex face of cisternae stacks is forming phase, while the inner concave one is maturing phase. The outer is convex and is called forming phase, inner is concave and is maturing phase. Cisternae breaks into vesicle from maturing phase. Cisternae move from outer to inner phase in a stack. Proteins synthesized in ribosome are converted to glycoprotein in Golgi apparatus. This is very important function that the protein which are synthesized in ribosomes are converted to glycoproteins in Golgi apparatus. Golgi complex functions in cell secretion. The main function of Golgi complex is that cell secretion. Both endoplasmic reticulum and Golgi transport cell secretion outside in the form of packing. Most important function of Golgi apparatus is to modify proteins and convert them into glycoprotein or glycolipids. Pancreatic enzymes are packed into granules by Golgi apparatus. Please remember these facts because these are very important. Now, lysosomes. Lysosomes differ from other organelles due to their morphology, means shape and structure. They were first isolated by Didieu in 1949. They are splitting bodies and engulf any foreign body and digest into simple form, means any foreign body can be engulfed by them and then it is converted or break into simple form. This eating process of cell is phagocytosis. Abundant in phagocytic cell, mean lysosomes are very abundant in phagocytic cell. Single membrane bound contain acid phosphatase and hydrolytic enzyme. These enzymes are synthesized on rough endoplasmic reticulum and processed in Golgi apparatus. This is also very important fact that where these enzymes are synthesized on rough endoplasmic reticulum and in Golgi apparatus processed. Processed enzymes budded off from Golgi are called primary lysosomes. These enzymes, which are synthesized on rough endoplasmic reticulum and then processed in Golgi apparatus, then budded off from Golgi, and these are called primary lysosomes. Primary lysosome, when fused with phagocytic vacuole, form secondary lysosomes. The primary lysosomes then converted or attached with phagocytic vacuoles, then called secondary lysosome. Lysosome which digest their own cellular part during starvation are autophagosomes, means that lysosomes digest their own parts. Then they are called as autophagosomes. Digestive vacuoles which perform digestion and autophagosomes which are self-eaten are called secondary lysosome. Autophagy or self-eating occurs for old or worn out parts of the eukaryotic cell like mitochondria. Lysosomic enzymes can also degenerate complete cell as during development. They also release enzyme for extracellular digestion. The disease caused due to accumulation of substances in cell are storage disease. These diseases are caused by mutation in lysosome enzymes. Glycogenosis type 2, liver and muscles are filled with glycogen. In glycogenosis type 2, the liver and muscles are filled with which enzyme? Glycogen. 20 type of diseases known which occur due to absence of lysosomic enzyme. One of the diseases is Tay-Sachs disease, which is caused by absence of lipid catabolic enzyme. Tay-Sachs disease, very important to remember that it is caused by absence of lipid catabolic enzyme. Which disease is caused by absence of lipid catabolic enzyme? Tay-Sachs disease. Or it may also be asked as Tay-Sachs disease is caused by absence of which enzyme? Accumulation of lipid in brain cell lead to mental retardation or even death may also occur. Paroxysome. Didev and co-worker isolated peroxisomes from liver cells rich in peroxidase, catalase, glycolic acid oxidase enzymes. They were named because of involvement in formation and decomposition of hydrogen peroxide H2O2. They are found both in plants and animal cells and are single membrane bounded. They are rich in hydrogen peroxidase and produce oxidase and catalase enzyme. Their diameter is 0.5 micrometer. They are also found in protozoa, yeast, and higher plants. This is important fact that where peroxisomes are found, they are found in protozoa, yeast, and higher plants. The next 10 topics will be in the upcoming video, which consists of chapter 4 second half complete concept and summary. If you like the video, please subscribe our channel and stay connected. Support it, really appreciate our efforts. Please like, comment, share and subscribe my channel. Keep watching. Take care of yourself. Allah Hafiz.